Speak about glycolipids. Well, if we remember that the cell membrane is composed of 3% carbohydrate in the cell membrane. So 3% carbs. So these carbohydrates, they're either bound to lipids or to proteins. So we'll talk about the component of carbohydrates which are bound to lipids, and those are called glycolipids. Now, so glycolipids are lipids with carbohydrate moieties attached to them, and usually they'll be oligosaccharides. That means they've got like between 2 to 8 or 10 little sugar moieties, individual moieties attached end to end over there. So these are your glycolipids, and they are usually present in large amounts abundantly in the brain. And there are of two types. One is gangliosides. Now, these gangliosides, they act as receptors for microbes. So sometimes, microbial entities like cholera or E. coli or the influenza virus, they actually play a role in attacking our body by sticking to these gangliosides. So, for example, cholera toxin or the heat labile enterotoxin, they call this the LE toxin from E. coli, or even the hemagglutinin from the influenza virus, they all bind to these gangliosides on your cell membrane. Okay, so it just happens to be one of the sites where they tend to bind to over there. And they help in the infecting of human beings. Unfortunately, this does happen sometimes. The microbes learn to attack and hurt people in smart ways. Then you have the cerebrosides. That's another type which is found in your brain, and they play a role also in the making up of the membrane in the brain. Now, we'll just talk about some of these glycolipids in the context of blood grouping. So if you remember with your major blood group systems, you have the ABO system, and there's the RH system. Let's leave the RH system for now, just ignore that for now. And I don't want to scratch that, it's an important thing. But ABO, we'll focus on the ABO blood group system. Now, you know that you have certain blood groups, like the A blood group, the B blood group, the AB blood group, and the O blood group. Now, what makes you get the A blood group? It's because you have an antigen called A antigen present over there, and that's why you have the A blood group. But you don't have any B antigens, so you have antibodies. I'm drawing it like this in this shape. Antibodies, A, B's, antibodies against B, because that's what you don't have in your DNA, coding and putting it on your red cells. And for the B blood group, you have the B antigen present on the cell membrane. So these antigens are present on the cell membrane, okay? And the antibodies are present in the plasma, okay? So on the membrane of the RBC, you got these antigens, and for the plasma, meaning in the plasma or serum, what you have is the antibodies floating. Which antibody do you have here? In B blood group, you have antibody against A antigen because it's not present in the body. In the AB blood group, AB system, you have the A and the B antigen present, but you do not have any antibodies, no antibodies over here. So these people can take blood from anybody. They're called universal recipients. They're around 4% of the population. Quite rare, actually, even lesser than 4%. Then you have the O blood group. These people don't have any antigens on the body, so no antigens. AGS for antigens, no antigens. But they have got antibodies against A and B. Against A and B. So that means that you cannot give them any blood from A, B, or AB blood group because it will cause a reaction. So, but these people can give their blood to other people. So they are universal donors. So these universal donors are the O blood group, and we'll talk in more detail about this when we go to blood grouping and Landsteiner's laws and all those other important topics, and how blood transfusion is done, and the problems of mismatched transfusion and RH incompatibility between mother and fetus and all that stuff. It's very interesting. Just remember that the antigens over here, these antigens A, B, AB, and even O has an antigen, is based on these glycolipids. At least all of these except the O has got, it's more of a protein rather than glycolipid. I'll explain now. Okay, so how do you get these blood groups, these A, B, AB, and O? You have these antigens over there. A antigen is present in A blood group, B antigen is present in B blood group, A and B are present in the AB blood group. So if this is your RBC, and you have cells which have this thing called the H antigen. So there is an antigen called H antigen. It's like it's a bunch of amino acids that come together and make this antigen called the H antigen. This makes you get O blood group. Okay, just remember, think of it like H for head as a mnemonic. Okay, now if this H antigen has got an A antigen stuck to it, think of it like a cap on top of a head. This is how I like to imagine this. So you have the A antigen. What is the A antigen? This is carbohydrate. This is all just, sorry about that, just carbohydrate that's present over here. So if the carbohydrate is A, now this is not an O blood group anymore. This became A blood group. So please remember, this is your cell membrane. It has got the H antigen in most people, almost everybody, and that makes it O if there's nothing on it. But if you add some carbohydrate on top, now it became the glycolipid, and this is your A antigen. Or if it is B carbohydrate, now what do you get? B blood group. And if you don't have any A or B, this is just H antigen, naked head, no cap on it, then you have got O blood group. Please remember this. Now, there are some people who don't even have H blood group, H antigen on them. These people are called Bombay blood group. What do we call them? Bombay. Because it was found in Bombay. Right? There are around 100 people in the world with this. Now, what's the problem with Bombay blood group? Look, these people never saw the H antigen in their life. So if you give them O blood, also, the O blood has got an antigen which the Bombay blood group people do not have. So what's going to happen? It will cause a mismatched transfusion. There will be a reaction, and it will cause damage to the tissue. There will be a type 2 hypersensitivity reaction, which is your mismatched blood transfusion reaction that's taking place. I'll emphasize on this when we go to that topic about what is type 1, type 2 hypersensitivity and all of that stuff. Okay, the point is no H antigen gives you Bombay blood group. So that will cause mismatched transfusion even if you give them O blood group in those people. So these are your glycolipids. Let's talk about the proteins a little bit. So there are two types of proteins in your cell membrane: integral proteins and peripheral proteins. The integral proteins are also usually called transmembrane proteins. Why is it the case? Because they you remember your fluid mosaic model? Yeah, these are your phospholipids. These transmembrane proteins traverse the entire length of the membrane, the entire width of the membrane, and so they come out on both sides. Can you see? Oh, here, so you can see them on both sides. So these transmembrane proteins are going to be visible on both sides, so they traverse the entire length, width of the membrane. So they're called transmembrane proteins. They are amphipathic. What does that mean? They have got hydrophobic and hydrophilic regions. Remember your cell membrane. Why is it lipid bilayer structure? If we go through the notes over here, that's because you have got parts of the molecules that make up this membrane which love water, which face the outer surface, which I've already explained, and you have parts that repel, which are repelled by water. Like in these phospholipid molecules, you see the fatty acid chains over here. They are facing each other because they are really uncomfortable in the water. But and same thing with these proteins over here. These proteins which you see drawn in green over here, they have parts that are exposed to water and parts that are close to the fatty acid chains and the hydrophobic water hating regions over there. So these are your hydrophobic regions, and the parts that are exposed are hydrophilic or water-loving regions. So they are amphipathic molecules. And I want you to know that mostly these are these transmembrane proteins, these integral proteins, they make up your transporters or ion channels, or they will make up some receptors which carry a signal. They sense some chemical from the outside, and they or a change in voltage somewhere, and they will pass the signal to the inside of the cell. So that is how they work. Usually they sense some ligand or some chemical which interacts on one side, and they pass a signal to the inside of the cell. So they are usually receptors, or they are transporters and carrier proteins and pumps which are responsible for cell membrane transport across the membrane, and that's usually what they end up being. And you should remember that the parts that cross the membrane, that are the hydrophobic regions, which you see over here. So you see there are the hydrophilic regions that are on the outside, and there's the hydrophobic region which is inside. The protein over here in the hydrophobic region is usually in the alpha helical configuration. What does that mean? In biochemistry, you would have studied that you have the alpha helix and the beta pleated structure of protein. So what happens is the outside will have its own shape, whatever, but the inside will have this helical shape, almost like your DNA. Okay, so that's how your cells are shaped. And then again, the hydrophilic region outside is shaped like so. So the alpha helical shape is what traverses the membrane, and it's hydrophobic, and it's more comfortable being in the middle of this phospholipid bilayer. In the third layer of the sandwich, if we look at the electron microscope appearance of the cell membrane, it looks like a sandwich with two thick layers on the outside, which is the polar heads of the phospholipids, and the thin fatty acyl chains being the peanut butter and jelly with the light part, the light colored appearance of the membrane in the middle. Peanut butter and jelly is not light. Anyway, so look at the receptors. There's something called G protein coupled receptor. G protein coupled receptor. I'll tell you what this is when we go to that topic on what do we say, cell intercellular communication over there, and this plays a role in the interaction of many hormones and chemical messengers, and we'll be going through that. It's a very important topic. Okay, just wait for it. So these G protein coupled receptors, they are also known as 7-pass receptors or serpentine receptors, because they cross the membrane 7 times. 1, 2, 3, 4, 5, 6, 7. You could see, and so this is your lipid bilayer, you could see over here, and you have these G protein coupled receptors interacting over here. Why do we call them G protein? Because a ligand will bind over here to the outer surface, and these receptors are bound to some other components on the inside, some other parts like alpha, beta, gamma in the big G protein, the heterotrimeric G protein type. Again, I'll go in more detail in that topic. Just remember that they bind to these proteins on the inside of the surface, and these proteins have the ability to bind to either GTP or GDP, depending on whether the receptor is bound to the ligand or not. So this GTP is bound to your alpha helic, to your alpha moiety over here, your alpha component or subunit, when you bind a chemical like a hormone or something to the receptor. And if there's no hormone or chemical over here, no ligand, then GDP binds to the alpha thing, and signals don't get transported. But if GTP binds, then signal gets passes through the cell. Just remember this for now. Now look, these 7-pass receptors, can you see they pass 7 times in and out of the cell membrane? So when you see the part that goes into the cell membrane, it's helical shaped. It's all helical shaped. Helical. Can you see that? This is what it is. Now, this is your hydrophobic and your hydrophilic regions over here that are present in your integral proteins. And how are they held in the membrane? It's because of hydrophobic interaction and van der Waals forces. Hydrophobic interaction and van der Waals forces. Proteins can be attached via lipid anchors also. Just be aware of this. And they get bound covalently to lipid molecules. And if we look at the outer leaflet, remember GPI? This was a very important type of component, which I already emphasized in the previous video. Well, so what happens is these proteins can be embedded to the outer leaflet via covalent bonding with the GPI molecules over there, okay, on the outer leaflet. For the inner leaflet, if we look at it, they can be bound to amino or via the amino or carboxy terminus, meaning you see your proteins have an amino end, and you make a little protein, and then you have a carboxy end over there in every protein. So the amino terminus can get bound to some component like your fatty acids in the inner leaflet surface by binding to fatty acids like myristate or palmitate. And the carboxy terminus over here can be bound via prenyl anchors to farnesyl or geranyl. So something it's quite a mouthful to remember, but that's how they interact and stick to the inner membrane. Outer membrane is outer leaflet, it is the GPI. Inner leaflet, it is the amino and carboxy terminus. Then you have the peripheral proteins. They bind by covalent bonds to cell membranes and by electrostatic and hydrogen bonds to integral proteins over there. So they're mainly for structural support, and they're more like supporting protein. They don't do a lot of transport across membrane. They can't, because they don't go through the entire width of the membrane. Right? And if we look at some examples of peripheral proteins, they are an example is a chemical called alpha spectrin. Or beta spectrin and N-chirin. So what is this? Well, if we remember our RBC, this shape of the RBC, it has something called the biconcave disc shape over here. If we look from the side, this is the way it looks, so it's like 2.2 microns on the periphery, and the center is around 1 micron in width over here, and this is around 7.2 microns in diameter, micrometers in diameter. So this is the appearance of your RBC. It's kind of like it's not a donut with a hole in the middle. It's like somebody tried to make a donut but didn't punch a hole in the middle. It just squeezed it but didn't make that hole completely. So what happens with your RBC biconcave shape over here is it is held in shape by some peripheral proteins, and these peripheral proteins are given the name alpha spectrin, CTRIN, beta spectrin, N-chirin, and band 3. And I will speak about this in more detail when we go to the chapter of elliptocytosis and ovalocytosis, which is a kind of anemia where your cells don't have this normal shape, because this cell membrane, it's got this spectrin over here as a structural scaffolding under the cell membrane, and it is held in place by N-chirin and band 3. So if any of these mutates, spectrin, which is the scaffolding, or N-chirin, which anchors this in place, you cannot maintain the biconcave shape, and instead you will get an oval or a round shape. So you get ovalocytosis or spherocytosis. Ovalocytosis can also be called elliptocytosis because it's shaped like an ellipse, elliptical shape. Or spherocytosis means it's just a proper circle over there. That's not good. Your cells RBCs won't survive long. They won't reach 120 days of life because of the abnormal shape. Why? Because look, I want you to understand that your RBCs are on average 7.2 microns in diameter, and they survive for 120 days. And what happens is they're supposed to go through small blood vessels in the spleen and certain other parts of the body. But especially in the spleen, the diameter of the vessel is only 3 microns. So what does that mean? That means that your RBC, when it goes to the spleen and passes through those vessels, it has to be very flexible, like a bag of You know those water balloons which you fill with water and throw on your friends and play a prank on them? It has a certain shape, but if you bend it, it will change its shape. That's what your RBCs are like. Or, if you come to places like in India or in Southeast Asia, when you buy curries or liquids, they actually pack it in plastic bags. They just put some rubber bands and put the curry or the coconut water or whatever you order, the fluid food items you order, in these bags. And you even get milk over here in plastic bags over here. So these plastic bags, they have a certain shape, but when you shape it, you put a little pressure, it will mold and it will change its shape. This is what your RBCs are. They're like bags of curry or coconut water, which you can purchase over here in Southeast Asia. And they pass through these like that. These 7.2 micron cells will be able to pass through these 3 micron vessels in the spleen. But if you have these mutations in the alpha spectrin, beta spectrin, ankyrin B, and the ankyrin and band 3 protein, then this structure will not be maintained. It will be spherical or elliptical, and they don't have that much flexibility. They get stuck over here in the splenic vessels, and the spleen is full of white cells that will just come and swallow anything that is blocking traffic in the blood vessels. So they just destroy your cells quickly. So spherocytosis, elliptocytosis. Okay, this is the problem. You undergo a kind of hemolytic anemia, meaning your red cells get destroyed before their time is up, because they cause a lot of problems. And so that's your spherocytosis and elliptocytosis, and the importance of the glycolipids. How does your cell membrane work, when it's injured? So if you have a small injury like a microneedle over there, you just make a very small hole with a microneedle. It rapidly reseals because there is this self-sealing process by hydrophobic interactions. Okay, and whenever it's a large break or macro injury, that's when some calcium-dependent processes come and play a role in holding things back in place. So that's beyond our scope right now. All what we need to know is this much for now.